well, before I start the speech, then I must say I did have exactly the same experience with my mother, and I can tell you that the most powerful experience that I had is to read a book which was called Insights of a Stroke, and that helped me to go through. So I invite you to continue writing that paper that may inspire people. So, colleagues, thank you very much. I would like first to extend my gratitude to the organizers of this conference, so the Flemish governments, in general, the Belgian presidency, and the Scienziano, because I realize, and I mean, even over the last 15 minutes, 20 minutes, colleagues and friends have told me the sheer amounts of work that was done yesterday. I've been asked by the organizers also to say a few words and essentially try to find a common line to what I wanted to talk to you about, and I do see that common line. So if yesterday we started to discuss, let's say, the genomic data infrastructure, which, for reasons that are related to my previous job, I know about, and I know about the legal, the organizational, and the technical underpinnings that are necessary to make that a reality, and how important it is to create such infrastructure to unlock, say, new knowledge. We moved from that, so the genomic data infrastructure, we start also to discuss applications of it and the role of industry, and the role of industry in creating partnership to promote innovation. We heard already let's say, the work that they're doing in the US in pushing forward the boundary of innovation. But we also included in the discussion the voice of the patients, in a way, so that we never forget for whom we are doing our work. Now, it is important because, I mean, I can tell you that I can testify. I've been working in DG Connect for quite some time on health, and now I work in DG Santé in a continuum that takes me from, say, the development of new technology to the adoptions and to the mise à disposition, if we work in French, of that technologies to patient. And that is a continuum that per se requires hard work, and we cannot do it in isolation. We need to do it together. And there are some elements that are important for that, including the creation of a regulation that is conducive to promote innovation and therefore adoption. And one of the pillars of that is the fact that we create through regulation, technology development, computing capacity, the way in which we package this into a framework that provides trust to citizens. So today, we are also going to develop further on the same analysis, and there are some subjects that are very close to my heart, including the digital twins and artificial intelligence. But we're also going to talk about, let's say, how in the complex biological systems we start to understand the diseases and what is the role of digital and these new technologies for that. We're also going to talk about ATMP, and so advanced therapy, medicinal product or gene therapy, which is also important. And by the way, I am also very honored to be working on health technologies assessment, which is to cover part of these technologies when it comes to, let's say, improving, the way in which we take these technologies to citizens. Now, the common thread, it is data and it is trust. That's why I'd like to focus a little bit on the European health data space, and I would like to do it from the perspective, say, where I am now, as someone who is working on the aspects of the regulation in itself. You may, I'm sure that you know, that there is a political agreement achieved on the European health data space regulation. We expect this to, let's say, enter into force by the end of this year. So late in the autumn, it will be published, and then it will become a reality. And this piece of regulation, which has been created in this collective spirit, I mean, not only between services of the European Commission, between DG Connect, Santé, but also with the people that will be using it, especially the researchers. I think that your voice has been heard in developing that regulation. Now, it will facilitate access to health data for citizens and professionals, and it will also facilitate the reuse of that data for research. It was very much needed. this regulation. It was very much needed. It has taken not a considerable amount of time to be negotiated. If you look in terms of negotiation, it went from 2022 to 2024, 2 years to negotiate a piece of paper which is more than 100 articles. You may understand that everybody was ready to discuss this because everybody saw the potential of that regulation. And everybody saw the potential of that regulation because when COVID came, we desperately needed access to that health data to solve the most pressing issues that we had at that time. So what we will do, in addition to creating the rights to citizens to access their health data, we will try to solve the interoperability issue, introducing in the regulation the European Electronic Health Record Exchange Format. What is this? It is essentially a mechanism that will allow us to transfer data from one source to another in a way that one that receives can actually unpack and understand what is there. Now, this is easy to say, but we did move forward, and I will tell you about that on what is already there. Now, the second aspect, which is very close to your community, is the fact that we not only open up the data to the citizens for their care, needs, but we also open up that data in a trustful way for research, for innovation, for policy making. Now, the regulation does a series of things. It first identifies the categories of data that can be used for secondary use. So it tells these are the categories of data that we need to work on, that they can be shared with the rules to ensure trust, to ensure security, to ensure privacy. And this covers a number of areas. I'm sure that you're already aware of that. But in addition to defining the categories, the regulations also define what is the purposes for which the processing of this data, can lead to sharing. And that includes research, it includes innovation, it includes policy making, statistics, But eventually, what is important is that access and sharing of health data shall be done in a way that is not detrimental to the individual, and it is very much valuable for the constituency that will use it for scientific purposes. So it's obvious that we, being the first huge experiment worldwide, it needed, to find a fine balance between these aspects. Now, how do you, once you know what are the categories and the purposes, how do we support, let's say, the sharing of data? That requires the establishment of, let's say, what we call the Health Data Access Body in the regulation, which is essentially the central networks where the researcher can go and ask one permit that will be valid in Europe. You define what is your project, you decide what is that you want to get out of the data, you tell them what data you are looking for, and that authority will be networked so that you get the permit to access that data. So it ensures, of course, that the rules are respected, and therefore the trust and the security of the entire environment, it is guaranteed. It's also very important that this health access body, they make transparent the rules and the conditions about the availability of that data. Some of that data may be anonymized, but some of that data may be pseudo-anonymized. So the way in which you access and reuse it must be very well defined, and that it's part of the permit that will be issued by the Health Data Access Body. Also, the data holders, they have to describe what are the data sets that they have at their disposals, and they have to make available that data if the Health Data Access Body requires to make that data available. Now, when it comes to use, and that is what the regulation will do, it will specify, let's say, the characteristics and the specifications of the secure environment that will need to be set up to analyze and work on that data. And by the way, that's also specify that, once the data users gets the data, they cannot share it with somebody else, as a way of proxy. That is very clear in the regulation. Now, the negotiation was very much focused on the rights of the individuals, and I'm pretty sure that most of you know that the final, let's say, political agreement includes the rights of opt-out to people for certain particular categories of data, and there are, let's say, also you will see that there will be provisions for addressing this at European level. Now, it's obvious that these provisions they aim to ensure that the needs of all data users are addressed, all of them. And, we know why we had to do that. Again, in my humble experience, I think that the fact that we went through our experience on COVID really highlighted the need for having such regulation. And we call it an enabling regulation because per se, well, it's important for the primary use. It's certainly important to guarantee the continuity of care when people move from one country to another or from one region to another region, because you will see that there will be spillover effect in terms of interoperability and access to data even within countries where perhaps a little bit of recommendation exists. So on primary use, it's obvious, I will benefit because it will in fact enable the roaming of health data in a way that is totally controlled by the users in a secure way. And for the secondary data, now that's what I would call in a way the Copernican revolution also for the regulation in itself, that this is the first time we put access to data for primary and secondary use in the same regulation. I guess, in my opinion, the fact that we went through our experience in COVID facilitated that discussions with the member states. Now, to be clear, this is a paradigm shift, but it will require time because it's not just about creating the right conditions from law, and also communication about it, but it also required setting up technological solutions that will allow the sharing of data. So if the things goes as planned and it will be enacted, later in the autumn, we will have the start of the let's say the obligations to start sharing that data as of 2028, let's say that we are not going to wait 2028 before we start implementing the European Health Data Space. It's so important that we start doing what we have to do right now, and so at the moment we are working not only to finalize the text for its adoption, but we are also working to create the underpinning technological infrastructure that will enable, number one, the transfer of data for continuity of care, primary data, my data, the mechanisms for me to access and control my health data and decide with whom I want to share, but also, importantly, the infrastructure that will allow you to request access to data, obtain a permit, obtain the data set, and do whatever you have to do for your research. Now, that means on our side that we start using projects for the health data infrastructures. This is how we are called, but we are also already supporting a project for the establishment of this health data access body, what we call the joint actions. For those who are familiar with U4Health program, they may know what it is as an instrument, as a funding instrument. For those that work for Europe, on Horizon Europe, they might not necessarily be so familiar with that. But technically speaking, what I wanted to say is that once we will have that framework in place, technological and regulatory, this will also, give a boost on further development of AI technologies, and certainly that they are in desperate need of accessing health data, especially if you want to tailor these foundational models in specific applications in the healthcare domain. So I think I want to close my intervention here by, first of all, expressing my gratitude for the invitation, because I believe that this is the audience that will certainly be the one who will, let's say, gain the more out of the regulations and infrastructure for secondary use of data. I'm very happy for that. But I'm also, to be very honest, a little bit selfish. I'm also very happy because this allows me, even if it is 6 months, to get in contact again physically with my colleagues from Luxembourg and from Brussels, that we are working together, and that I'm sorry if I say so, but it's also part of it's part of the package. So I thank you for the invitation. I hope that you will all benefit from the European Health Data Space. I hope that you will share the message of the importance of the European Health Data Space when this will become a regulation. And finally, I hope that you will be able to develop the technologies that our citizens are in need of. Thank you very much again.
Good morning, everybody. Indeed, I'm Bernard Thienpont. I'm co-founder of the Leuven Institute for Single-Cell Omics. And today I'll be talking to you about single-cell and spatial multi-omics and about the opportunities that these technologies are giving to transform precision medicine, but also about some of the hurdles that come with these technologies. It's very much applications talk. I apologize. I don't have a lot of experience in policy making. So let's start, if my slides want to continue. Yeah, there we are. So as we know, as many of you know undoubtedly, the human body is composed of about 100 billion cells. Each of these cells expresses their own gene expression program, and together these gene expression programs encode tissue and organ function, and all these organ functions ultimately make us the human beings that we are today. Typically, to study these tissues, we look at these in bulk. This is how it was done historically, and so then we can see that gene 1 is lowly expressed and gene 3 is highly expressed. However, with the advancement of single-cell multi-omics, now we can look at each individual cell in isolation. And we can learn, for example, that the blue cells express only the blue cells express gene 1, only the pink cells express gene 2, and all the cells express gene 3. For the moment, we don't know how many cell types there are in the human body, and this is the task that was set out in the Human Cell Atlas. This is a project that is in grandeur, similar to the Human Genome Project, so that aims to decipher all the different cell types that are present in the human body. Apart from looking at cells which are terminally differentiated, we can also look at cells which are en route to be differentiated, and we can put all these different cells in a trajectory and study how differentiation occurs by putting them, sort of say, in pseudo-time. A clear application of this is the study of how a single fertilized totipotent zygote develops into a fully fledged human adult, how gene expression programs in these adults change as we are aging, and how these programs are perturbed in diseases. And this can learn us something about novel treatment options or novel biomarkers for diseases, as I will show in the next couple of slides. Apart from looking at gene expression, we can also look at the single cell level at the genome of these cells, the epigenome that controls expression of these genes, but also at the proteins and the metabolites that are the products of these transcripts. And then finally, with the advent of spatial multi-omics, we can not only look at each of these cells in isolation, but we can also look at the gene expression programs of these cells in their native tissue context, so in the tissue as it is present in the body. And this can help us decipher functional cell niches and cell-cell interactions, so how are cells talking to each other physically in the human body. For those of you that are interested in this topic, we published recently a review on how these technologies are revolutionizing our biomedical field. Some of these technologies were pioneered in Leuven. I've highlighted them here. And as you can see, as time progresses, more and more of these technologies have become available, and they either profile more modalities from the same cell, or they allow us to profile more cells at the same time. This increase in throughput, so allowing us to profile more cells at the same time, was really a technology revolution. So while initially we picked individual cells, put them in a microvesicle and analyzed them individually, increases in throughput have been brought about by developments in droplet microfluidics and single-cell combinatorial indexing. And for droplet microfluidics, I've added a small movie here. So you're looking here at small channels, which are the width of more or less a human hair. And here, water is pushed in an oil solution. And by pushing this water in an oil solution, we can create very small vesicles, picoliters in size, that allow us to study, to compartmentalize cells and to study them in isolation from each other. I will now highlight 3 different applications of these novel technologies and how they can help us achieve precision medicine. They come from 3 very different fields. They're all examples from my own research. The first example is lung tumors. As some of you may know, lung tumors consist of cancer cells, but these cancer cells are typically embedded in a matrix of other cell types that provide support. We have immune cells, and we have, for example, endothelial cells that line the blood vessels that supply blood and nutrients to these tumors. Together, they form a very complex cellular ecosystem, but at the time when we started our study, we knew a dozen, maybe 20 of these different cell types, but we did not know whether these were indeed all the different cell types that are present in a lung tumor. We also did not know whether they form uniform populations. Is every endothelial cell the same, or do we have multiple types of endothelial cells? And is an endothelial cell in the tumor, is it the same endothelial cell as one we find in the normal tissue? Right? So to study this, we took tissues from patients that underwent lung lobe resection with curative intent. We took these tissue samples, we dissociated them into individual cells, we analyzed them using these miniaturized assays, and the output of this experiment is shown on this slide. This is a very complex slide, I apologize, but every dot that you see in each of these panels is a single cell, and the closer these dots are to each other, the more similar these cells are to each other. I've also color-coded each of these cells for the expression of specific genes, specific marker genes, and this allows us, for example, to identify which of these cells are the endothelial cells. We can then look more closely at these endothelial cells. We can isolate them specifically. There are about 1,600 that we identified. And when we look, we see that, in fact, there is not one type of endothelial cell. We could identify 6 different types. There's one that we knew. These are the lymphatic endothelial cells, but all the other ones were blood endothelial cells, and we did not really know what was the difference between both. As you can appreciate, there's also striking differences between endothelial cells which are in the normal lung and endothelial cells which are in the tumor. To understand these differences, we looked at the pathways that are activated in the tumor versus the normal lung. And you can see that, for example, the inflammatory response is strikingly downregulated in these endothelial cells. And this is surprising because a tumor is normally a highly inflamed tissue. When we zoom in into which genes are actually responsible for this downregulation of this pathway, we noted that they were mainly involved in antigen presentation and in immune cell homing. So normally when a tissue is inflamed, there's a signal that says, immune cells need to come into this tissue to repair the inflammation. And here we see that in tumors, even though they are inflamed, this pathway is downregulated, and these immune cells actually contribute to the immunosuppression that is needed for the tumor to survive. This is in line with earlier observations in mouse models, and also provided a rationale for combining anti-angiogenic therapies and immunotherapies in patients, and the clinical trials for those are ongoing now. In total, rather than having 20, we now identified 52 different cell types or cell states in these tumors, and these cell types are not simply bystanders. This is clear when we look at how the abundance of these novel cell types, for example, influences patient survival. Right? Some of these are associated with a good prognosis, and others, when they are highly abundant, are associated with a poor prognosis. These analyses also allow us to identify which genes are particularly expressed in activated immune cells, and this allowed us to identify, for example, novel immunotherapy targets. In the top 30 genes in these activated immune cells, 3 were known immune checkpoint inhibitors, and 4 are further down the list. Some of these immune checkpoint inhibitors are now either approved immunotherapy targets or they're in advanced phase 3 stages. So, to summarize this first example, single-cell omics can provide novel insights in disease processes. It can provide biomarkers for patient prognosis and targets for therapy. As you appreciate, this field is highly interdisciplinary. It requires the merging of biomedicine, engineering, and bioinformatics. And these are typically high-risk, high-gain projects for which current funding schemes are not always perfectly adapted. Another example is shown on this slide. So here we're not only looking at the gene expression, we're looking at from a single cell, the gene expression, the chromatin accessibility, and the DNA methylation profile. By sorting cells in individual wells of a 96 well plate and separating the messenger RNA from the genome, we can study the transcriptome of that cell, but also the DNA methylation profile of that same cell. This allows us to correlate in a single cell gene expression and DNA methylation, but also allows us to identify specific DNA methylation signatures for specific cell types. This is important, for example, for a project where we're interested in cell type-specific DNA methylation in the placenta. Why were we interested in DNA methylation in the placenta, you may ask? Well, because in talking to obstetricians and gynecologists, they expressed a particular frustration with preeclampsia. This is a disorder that is characterized by new-onset hypertension and proteinuria. These symptoms occur after 20 weeks of gestation, and there is a drug that can prevent this disease from occurring, but that drug needs to be administered well before the onset of symptoms. This is a major cause for maternal and fetal morbidity and is thought to occur because of placentation defects that occur very early in gestation. So to identify these placentation defects, we needed DNA methylation signatures from the placenta very early on. So we needed improved tools for early prediction. To cut a very long story short, we looked at cell-free DNA methylation in the maternal plasma, so liquid biopsies from the mother obtained at 12 weeks of gestation. And here we can identify placental DNA. We can study this placental DNA by using tailored technologies. So here you see, for example, a locus that is lowly methylated in blood, highly methylated in the placenta, and shows an intermediate methylation pattern in the cfDNA. So we can detect these DNA methylation differences in this maternal plasma. And then we studied cfDNA samples that were obtained around 12 weeks of gestation, so well before the onset of symptoms. We compared patients that would go on to develop preeclampsia with those that would not go on to develop preeclampsia, and this revealed a DNA methylation signature that helped us to predict which women are at high risk of developing preeclampsia and which are not. This signature was also validated in an independent cohort. As you can see here, we can achieve accurate prediction of early onset preeclampsia risk. This was a study that really sparked a lot of interest in the field. I think it's the first time in my life that I appeared in a journal in India also. And we are currently working hard to translate this proof of concept into a clinically applicable test. So using single-cell omics, we can identify novel, minimally invasive predictive biomarkers. But these DNA methylation atlases that I need for these tests are unfortunately for the moment scarce, and there's also ethical legal constraints that are unfortunately slowing progress. And then the last example that I want to highlight, this is very much work in progress, is one where we look at Parkinson's disease. Parkinson's disease is a disease that unfortunately we don't really know why people develop Parkinson's disease. So to try and understand why people develop Parkinson's disease, we set up a study in collaboration with the Aligning Science Across Parkinson's Consortium to try to understand genome-wide association studies and the hits that are identified there. So we are combining long-read whole genome sequencing with single-cell and spatial multiomics. And we try to answer two different questions. On the one hand, how are these risk loci perturbing gene expression in Parkinson patients? And then also there's this Bragg hypothesis, which is a very interesting hypothesis that suggests that Parkinson is in fact a disease that originates in the gut and is subsequently seeded into the brain. So we are also studying this. For this, we're using single-cell RNA and ATAC sequencing together with colleagues from Leuven and London. We looked at 3 different tissue types. So a control brain tissue, the substantia nigra, where Parkinson's is originating, and the ascending colon, where the disease perhaps originates. For this, we're looking at over 500 different patients, 750 samples, and we're analyzing over 3 million nuclei, both for gene expression and for chromatin accessibility. Again, this is very much work in progress, but some of the interesting hits that we are identifying there, we're trying to validate using spatial transcriptomics. And here we are trying to see whether cells which are changed in Parkinson's disease patients, whether they are changed in location in the gut, and whether cells that are interacting with these cells, whether they are also changed in Parkinson's disease patients. This, of course, requires advanced bioinformatics tools. We are developing concrete autoencoders to translate the single-cell transcriptomic data to this spatial transcriptomic data. So perhaps this latest project can provide novel insights into disease etiopathogenesis. It's clear that these types of projects have very strong computational demands. Spatial transcriptomics generates terabytes or even petabytes of data. And as we are progressing, I don't know if anybody has looked this up already, but we are getting towards exabytes of data from these types of experiments. So to summarize, to achieve precision medicine, we need to merge different fields. This will allow the study of cell biology in developing, aging, healthy, and diseased tissues. And this can be applied across a range of different disciplines. I've showed you tumor biology, neurodegeneration, pregnancy complications. And so to allow for this, we've recently founded the Leuven Institute for Single-Cell Omics. And this brings together all these different expertises with one common goal, clinical translation of our findings to help ultimately the patients. So we aim to unravel the etiopathogenesis of diseases, to understand drug mode of action, to identify new biomarkers, druggable targets, and ultimately to guide precision medicine. These technologies are not only valuable for discovery, they can also be clinically applied, and we are trying to push the field there. We are collaborating with industry and academia for training, and then most importantly also, we are aligning with industry to devise novel clinical trials that can help us to achieve our vision. And so with this, I would like to end. We're hiring if anybody is interested. And do feel free to reach out if you have any further questions, and I'd be happy to take any questions you may have now. Thank you.